So the very unique approach which our group is focused on exploring then is a very large scale, very unbiased, and I think that's really key to what we're trying to explore. Where the goal for this originally was to take an enhancer library with a random number of permutations in this. So in this case, we chose about a 2% fixed mutation rate. And really the reason we focus on the 2% is it's known that there's a very similar distance in terms of DNA sequence turnover that you get between wild and natural, or different natural flies in these closely related species. And we'll kind of return to some of this as we come towards the tail end of our talk. And so what we actually ended up doing then for this particular pipeline, which I'll walk you through now, is GenScript, who's been very good collaborators with us for the past, I'd say at this point almost a decade, is that they built us this very nice enhancer library of a very nice fixed mutation frequency around 2%. And then what we did here is we created about 1,000 independent lines of flies. And then I'm going to walk you through how we built this very high throughput embryology techniques, which is the really centerpiece of this is a lot of the robotics that allows us to handle all of this in a very automated fashion. And coupled with then the image analysis pipelines, that allow us then to relate changes in DNA sequence, right? So these very simple genotypic inputs to these very discrete outputs that we can see during development.